The goal of this procedure is to determine the mitochondrial membrane potential and levels of reactive oxygen species in primary cortical neurons using the fluorescent probes TMRM and H2-DCF-DA respectively. This is accomplished by first preparing stock and working concentrations of the TMRM and H2-DCF-DA. The second step is to incubate the cortical neurons with TMRM and H2-DCF-DA at room temperature for 45 minutes in Rhodes buffer. The third step is to image the fluorescence intensity of TMRM and DCF in live cortical neurons using 515, 570 nanometer and 488, 515 nanometer excitation emission respectively. The changes in TMRM fluorescence will be monitored in the presence of the mitochondrial uncoupler FCCP, while the changes in DCF fluorescence will be monitored in the presence of hydrogen peroxide. The final step of this procedure is the collection, normalization, and analysis of data. Ultimately, results are obtained by plotting a graph that shows the relative levels of TMRM and DCF fluorescence intensity before and after FCCP and hydrogen peroxide treatment, respectively.
Hello, I'm Joanna Bakowska. Welcome to my lab at Loyola University Chicago. Today we will show you how to measure mitochondrial membrane potential and reactive oxygen species in rat cortical neurons. The procedure will be demonstrated by Dinesh Joshi.
Hi, I'm Dinesh Joshi. Using this procedure, mitochondrial membrane potential can be measured at single mitochondrion level and reactive oxygen species at single cell level. So let's get started.
Prepare a 10 millimolar stock of TMRM by dissolving 5 mg of TMRM in 1 milliliter of anhydrous dimethyl sulfoxide. Vortex for 1 minute. Then make aliquots. Store the aliquots at -20 degrees Celsius, protect from light, and use within 1 month. Next, prepare a 10 millimolar stock of H2-DCF-DA by dissolving 4.87 mg of H2-DCF-DA in 1 milliliter of anhydrous dimethyl sulfoxide. Similarly, vortex for 1 minute. Then, make aliquots and store them at -20 degrees Celsius. Protect from light and use within 1 week. To load the rat cortical neurons with TMRM, first, wash the cultured neurons 3 times with Rhodes buffer. Then, prepare 20 nanomolar TMRM by diluting the 10 millimolar TMRM stock 1000 times in TB. Then add 2 microliters of the diluted TMRM per 1 milliliter of TB. Incubate the neurons with TMRM for 45 minutes in the dark at room temperature. After 45 minutes, mount the culture dish on the stage of the microscope and start imaging. To load the rat cortical neurons with H2-DCF-DA, wash the cultured neurons 3 times with TB. Next, prepare 2 micromolar of H2-DCF-DA by diluting the 10 millimolar H2-DCF-DA stock 10 times in TB, and then add 2 microliters of diluted H2-DCF-DA per 1 milliliter of TB. Then, incubate the neurons with H2-DCF-DA for 45 minutes in the dark at room temperature. After 45 minutes, wash the neurons 4 times with TB to remove excess fluorescent indicator before obtaining images. To perform live imaging of neurons incubated with TMRM, confocal laser scanning microscopy with the application of live time series program is used. Apply low resolution and attenuated laser power to minimize the time needed to obtain images and to avoid photo bleaching. Next, adjust the focus of the mounted neurons loaded with TMRM using reflected light. Examine the TMRM fluorescence by illumination at 514 nanometers and detection at 570 nanometers. Set the detection gain of the camera just below the saturation level. Once all parameters, which include resolution, laser power, detection gain of the camera, and time-lapse interval to obtain images are set, do not change these settings between experiments. Next, change the field. Start collecting images. To test changes in delta psi m, stimulus such as 1 micromolar of FCCP or 2 microgram per milliliter of oligomycin can be applied, which will significantly depolarize or hyperpolarize the mitochondrial membrane potential, respectively. These changes will be reflected by a decrease in TMRM fluorescence intensity compared with the baseline fluorescence intensity in the case of FCCP, or an increase in TMRM fluorescence intensity in the case of oligomycin. To perform live imaging of neurons incubated with H2 DCFDA, first mount the culture dish on the stage of a microscope. Adjust the focus of the cells using reflected light. Examine the DCF fluorescence by excitation at 488 nanometers and emission at 515 nanometers. Next, adjust the laser power to 5 to 7%, detector gain, and resolution of 256 by 256. Do not change these settings between experiments. Then, set the frequency for obtaining live images using the time series program. Select a new field and start acquiring images. To detect changes in ROS levels, treat cells with 100 to 200 micromolar of hydrogen peroxide. This will be reflected by an increase in DCF fluorescence intensity compared with baseline level. Use the region of interest tool from the LSM program to select the areas. Then, select ROIs from mitochondrial regions or ROIs from the entire cell body in image cells to measure the fluorescence intensities of TMRM or ROS respectively. Select regions next to the cells to calculate the background fluorescence intensity. Take several measurements and to calculate the average background intensity. Subtract the average background fluorescence intensity from average fluorescence intensities of regions of interest in each cell for each time point using Microsoft Excel. After subtracting background intensity, normalize the TMRM or DCF fluorescence intensity to the baseline fluorescence using this formula. Then, use the Sigma plot program to generate the plot showing the changes in fluorescence intensity over time. Here is an example of a fluorescence image of rat cortical neurons incubated with TMRM. The addition of FCCP, a mitochondrial uncoupler, leads to mitochondrial depolarization and a loss of TMRM fluorescence intensity. The baseline TMRM fluorescence level remains stable before the addition of FCCP. Quantitative analysis of TMRM fluorescence changes over time shows a significant decrease in TMRM fluorescence after addition of FCCP. Here is an example of a fluorescence image of rat cortical neurons loaded with DCF. The baseline DCF fluorescence level remains unchanged in the first 120 seconds before application of hydrogen peroxide. The addition of hydrogen peroxide results in an increase in DCF fluorescence intensity in the cell bodies. Time-lapse measurements of DCF fluorescence show its steady levels, which have increased after hydrogen peroxide treatment.
In this procedure, it is important to remember to use a low laser power and fast scan speed to avoid the artifacts from photobleaching and phototoxicity. Have a good luck with your experiment.